Hi everyone, today we'll be discussing the method for analyzing viral components. The first step is purification. So you would take the original sample, the original virus sample which you have isolated based on the disease syndrome, and then you would run either a plaque assay and then select the virus from a single plaque to grow into the stock, or you would limit the dilution of the original sample and just use that, or you could isolate the viral genes and then insert it into plasmid to generate a molecular clone. Usually this third option is the preferred option. Then you would disrupt the cells either by using freezing and thawing of 2 to 3 cycles. The crystals will disrupt the cells. This is safe, especially for non-enveloped viruses. You can also use non-ionic detergents such as Triton X100 or Nonidet P40 because it will dissolve the cytoplasmic membrane but not the nuclear membrane, releasing the cytoplasmic viruses. Or you can use a homogenizer like blender, which has rapid blades to disrupt membrane and cause vesicle formation, causing the rough endoplasmic reticulum to form into microsomes and disrupting the nuclear membrane, so you get DNA and chromatin forming a very viscous solution. For analysis of this viscous solution, you need to use centrifugation. Firstly, you use low-speed centrifugation to separate the dense nuclei and large cell fragments from the smaller organelles, ribosomes, and viruses in the supernatant fluid. Then, taking this supernatant fluid, you run it under ultracentrifugation, which is very fast because it is made from strong titanium alloys, sealed with a vacuum and has a controlled temperature. And then in the end, you would get virus particles left in the pellet and supernatant fluid containing ribosomes and proteins which are discarded. Then taking the virus particle pellet, you can separate it into fractions for analysis and place it into a buffer solution, putting it through density gradient centrifugation, separating it based on size, where larger sediments fall faster in rate zonal density gradient centrifugation, and this is done using a sucrose medium, or you can use equilibrium isopycnic density gradient centrifugation, where you separate it based on density and the denser cells fall faster. Using this method, you can collect several fractions, and the fractions can then be further analyzed using SDS gel electrophoresis and immunoblotting to look at the specific peptides and proteins within the fractions, or you can use mass spectrophotometry to look at the relative abundance and post-translational changes of the proteins. And for if you're looking at infected cells, then you would see both non-structural and structural proteins. But if you're looking at the virus itself, then you would only be looking at the virus structural proteins. Lastly, you might want to look at the genome and determine whether or not it is an RNA or a DNA virus. So the way you do this is by using radioactive compounds, 14-carbon-thymidine and hydrogen-3 uracil. So these are basically nucleic acid bases conjugated to a radioactive compound, and which can be used by infected cells to produce virions. You can then isolate the virions and use a scintillation counter to find the amount of radioactive compound, 14 carbon or 3 hydrogen, within the virion. If it has more 14 carbon, it is obviously a DNA virion, but if it has more RNA, then it is an RNA virion. And then you can figure out whether or not it is single-stranded RNA by seeing whether if it gets digested by RNase A or T1. If it does, it is single-stranded RNA, because this compound doesn't digest double-stranded RNA, only single-stranded. and then you can use TCA to precipitate the remaining nucleic acid for analysis. Thank you so much for listening. Have a good day.